Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Nabil M. Malaki. My student ID is 20211222987, and my class is A4AS1202A. I will explain about aerobic, respiration, and fermentation. Okay. The introduction. Aerobic respiration is a type of cellular respiration which needed oxygen in variety of molecule by cellular respiration. The molecule in the form of fat and protein can enter the cellular metabolic pathway to break down sugar to make ATP. Energy can be extracted from respiration at appropriate stage of metabolic pathway to the broken down and reproduce ATP. As for aerobic respiration, around 30 or 32 ATP are produced in the matrix of mitochondria from the synthesis of glucose. Other type of cellular respiration is fermentation. It is also known as anaerobic respiration, where sugar breakdown pathway that ATP produced and does not need oxygen. Fermentation first begin with glycolysis in cytoplasm, but it concludes reaction that take place in cytoplasm which different from aerobic respiration. But in this reaction, no electron move through electron chain, so no ATP is produced. Other than that, instead of oxygen as the electron carrier, fermentation process use substrate level phosphorylation to generate ATP. When the puree produced, it is converted into different type of molecule on base fermentation. Okay, the content. The first question of the study case is a summary equation to cell respiration relate with redox reaction. Okay, this is the equation that is glucose C6H12O6 plus with oxygen 6O2, and it will produce carbon dioxide 6CO2 plus water 6H2O plus with ATP, that is energy. Okay. When the carbon hydrogen bonds of glucose are broken, electrons are begin transfer to oxygen. Transfer of electrons during chemical reaction release when stored in organic molecule. The released energy is ultimately used to synthesize ATP. This is the overall reaction of cellular respiration. This is the redox reaction. As you can see, this is the glucose. Glucose will oxidize to carbon dioxide, and the glucose will lose electron and hydrogen, and the oxygen will reduce to water, and if and oxygen will get electron and hydrogen. Okay, question number 2. Describe all stage in aerobic respiration and anaerobic respiration. Aerobic and anaerobic respiration stage. First, the first stage of aerobic and anaerobic respiration is glycolysis. In this glycolysis, the glucose molecule breaks down in two or three carbon compound, pyruvic acid. So, in the second stage, the pyruvic acid undergoes incomplete oxidation. For example, fermentation. The in the incomplete oxidation is pyruvic acid, ethanol, and lactic acid. For the anaerobic respiration, there's only two stages, that glycolysis and fermentation. Okay. For aerobic respiration, there are more. There is critical and electron transport system or change. Okay, we will proceed to the number 3. But in aerobic respiration, the pyruvic acid is acetylated before enter into the Krebs cycle. The next stage is Krebs cycle completed in the matrix of mitochondria. Okay, the last stage is electron transport system completely in the inner mitochondria membrane. Water is formed by utilize, utilization of oxygen and hydrogen supply from the NADH and FADH. Okay, the question number 3. State major difference between aerobic respiration and fermentation. First, one of the major difference between aerobic respiration and fermentation is presence of oxygen presence. In aerobic respiration, oxygen is needed in the process. When there is the presence of oxygen, aerobic will enter the mitochondria where the oxidation of glucose is completed. Ferment, fermentation oxidize glucose to produce lactase or ethanol without use oxygen. The next, the amount of energy in terms of ATP produced both in aerobic respiration and fermentation are different from each other. When there is no presence of electron transfer change, the pyruvic become unable. Thus, fermentation process occur which yields two molecules of ATP. However, in aerobic respiration, pyruvate is completely oxidized. NADH and FADH to carry the electron which contain most of chemical energy across the electron transfer chain. Thus, 32 or 30 molecule of ATP managed to be produced from this process. Okay, next is question number 4. What is the significance of NAD plus in the fermentation and cell preparation? Respiration. Okay, my answer is NAD plus is so important to both fermentation and cell respiration because NAD plus need to maintaining and continue glycolysis process in the cell. In fermentation, electron transport chain does not happen because there is no oxygen that act as final acceptor to pull and receive the electron. So APT cannot be produced. For that reason, NAD plus will regenerate on its own by undergoes reduction to become NADH and will be oxidized back to NAD plus. Question number 5. What does Stanley need to do to redesign his original experiment? In Stanley's original experiment, he did not close the container of the yeast. In this way, the process of fermentation did not happen. To redesign his original experiment, Stanley needed to close the container of the yeast to prevent oxygen from entering the container and react with the yeast. When the yeast does not react with oxygen, fermentation will occur. The cytosol of the yeast cell and it will break down the sugar into alcohol and carbon dioxide. So, Stanley need to use glass with the lid to create vacuum. Okay, the conclusion. The conclusion is each living being experiences cellular respiration, either an, an anaerobic or aerobic respiration to gain energy in ATP. Anaerobic respiration used inorganic molecule as their final electron acceptor that not require oxygen, whereas aerobic respiration requires oxygen as the final electron acceptor to create ATP. There, there are few stages in aerobic respiration, which are glycolysis, degradation, Krebs cycle, and electron transfer chain. Although aerobic respiration also have had add up to 30 or 32 ATP create, are created from all the stage. Meanwhile, in anaerobic, there are too short of respiration, which are alcoholic alcoholic fermentation and lactic acid fermentation. As it are glycolysis and one special reaction that regenerate NAD plus, which can be reused by glycolysis that happen in anaerobic respiration. In alcoholic fermentation, pyrothef is reduced to ethanol. Meanwhile, in lactic acid fermentation, pyrothef is reduced to lactate. 2 ATP is discharged by anaerobic respiration. This energy is discharged and after that used to do work such as cell reproduction, separation, and separation of chromosome and cell repair. Okay, this is the reference that I got, and thank you.